Hello friends, today we'll discuss about something known as assisted reproductive technologies. Assisted reproductive technologies simply speaks about another type of human intervention in the field of reproduction. That is, it is, as the name indicates, it is a kind of help or human intervention, artificial or assisted kind of reproductive technologies which help the infertile couples to conceive, to have children. So it is a general term referring to methods used to acquire pregnancy by artificial or partially artificial means. There are several methods have used to modify or stimulate the process of human reproduction and to treat severe cases of infertility both in males and females. Are new techniques that have developed over years to help infertile couples to have children. There are so many techniques. The one very common which everyone knows is IVF, in vitro fertilization. There are other types like embryo transfer, artificial insemination, gamete intrafallopian transfer, zygote intrafallopian transfer ZIFT, intracytoplasmic sperm injection ICSI. So we'll start with IVF. So this is a kind of pictogram which shows the basics mechanism behind IVF. Here what is happening is that the mature egg is harvested from the female, is taken from the female, and at the same time through natural ejaculation or from the sperm banks, the sperm is also collected from the male. So both the egg and the sperm, the gametes, that is the egg female gamete and the sperm male gamete, are fused in a test tube outside the body in the lab. So we call that as in vitro. In vivo means inside the body, in vitro means outside. So here, since the fertilization, the main fusion happens outside the body in the lab, we call this technique as in vitro fertilization. Once it is fertilized, a single cell embryo is obtained, and then further it will be kept in the media to undergo divisions, and once it is a proper stage is reached, it is transferred into the mother's body. And here we could see that IVF, there are some procedures included in the IVF, and IVF is a method which is specially being used to treat infertility caused by oligospermia, that is the number of sperm is less, or sperm antibodies are present when infertility is caused because of the presence of sperm antibodies, or there is some problems with tubal disinfection. That is, tubal function dysfunction means if there is some block in the transfer of sperm from the male's body to the female's body, or if there is some problems of endometriosis which you may be knowing, as well as if there is some unexplained infertility. So this is a solution for infertility caused, most of the infertility caused by the above said reasons. So now we'll just move on with the procedure, step by step, how this is being carried out. So for IVF to be carried out, and we can see that this IVF becomes the basis of the other type of assisted reproductive techniques we'll be discussing later on. So IVF is a common procedure which is followed. Later on it is clubbed with other methods too. So here in IVF, the first is to stimulate the ovary. If there is a problem with the female, if she is not capable of producing a proper mature egg, or if we don't want to take any more risk, or we cannot wait for a long, because already it has been identified that there is a infertility problem between the couple. Here when we go for IVF, we don't need to take any risk. So the first method is we go for a controlled ovarian stimulation. Here what happens is that the ovaries are stimulated or induced to produce more number of egg. In natural condition, only a single egg is produced or is being matured, and it is released from one side of the of the ovaries but then here we stimulate the ovaries and it is in a controlled manner so that more than one mature eggs are being produced. So alone here we will be giving some stimulating hormones or drugs like clomiphene plus gonadotropins or gonadotropins alone can be used. Anything in combination can be used or it can be released. And here we can see once they are being stimulated by drugs, we can see that it have some drawbacks also, because sometimes ovarian hyperstimulation syndromes can also be seen, like there may be overbleeding, there may be some other abdomen pain, and there can be some issues. Even if we go for this IVF, one of the drawback with the IVF is that it may lead to multiple pregnancy. So that is a problem. Then second step, when I say it is here oocyte retrieval. Now we know that more than one egg will be produced, that for sure, because we have already induced, we have overstimulated it. Super ovulation have been happened. Now the next step is oocyte retrieval. About 34 hours after this human chorionic gonadotropin is given, if in this treatment of control ovarian stimulation, we also give HCG in the later stages for the proper maturation and ovulation. And after this maturation, after that treatment, after 34 hours, we can retrieve by direct needle puncture of the follicle, and usually transvaginal. It is done through the vagina from the lower part of the body using an ultrasound guidance, and very little chances are there when laparoscopy is being used. Now next it is the fertilization. Once the oocyte are inseminated in vitro, that is oocyte is taken, and also artificial insemination, not artificial insemination, ejaculation is being done, and the semen is mixed with the oocyte. The semen samples are normally typically washed several times with tissue culture medium and is concentrated with motile sperm, which are then added to the medium containing the oocyte. That means once the semen is collected, it is further treated. It is treated with several washes of the cultural medium, and then the sperm only, we'll make sure that the motile sperm, there is a concentrated collection of motile sperm, and that is mixed with the oocyte. After which we can see that it can be transferred. Once the embryo is once the fertilization is been complete, we can see that the next is the embryo culture. Once the single cell is formed, it further is allowed in the media to undergo further multiplication. About 2 to 5 days it is kept in the media itself, after which once the embryo is developed, it is transferred. Only one or a few of the resulting embryo are transferred into the uterine cavity, and this reduces the chance of multi-fetal pregnancy. That means, I already told you, the doctors don't take risk. They simply don't give a chance with a single embryo. They go for more than one embryo, so that if one fails, the other will develop. That's why most cases IVF result in multiple pregnancy. Most probably twins are being produced, or triplets, or there are quadrants being produced. There are results even there are been reports where 12 babies at a time, or 6 at a time have been produced because of even. And you might have heard about the famous choreographer Farah Khan. After IVF, she had 3 babies. Karan Johar, even Shah Rukh Khan and the Gauri Khan, they also opted IVF. So there are so many examples like that, and we can see that some or all embryos, especially if women are at a high risk of ovarian hyperstimulation syndrome and all that, may be frozen in liquid nitrogen for transfer in a subsequent cycle. So because it is kept in nitrogen, and it is if one cycle fails, that same frozen embryo is used in the next cycle. So there is an increasing tendency to place only one embryo at each transfer and to freeze the remaining embryos for use in subsequent cycle if pregnancy does not happens with the first risk case. So that is the case, and we can see that so that is the common steps we opt for IVF. Now we will just come back. So here you could see the first step is the super ovulation, or the it is being stimulated to produce more egg at a time, and it is second step is to take it out from the ovary. It is retrieved, the embryo, the oocyte is retrieved, and the third step you could see it has been mixed with the motile sperms, and it is kept for 2 to some days, 24 hours to 2 days, and after it has been 2 to 5 days it can be kept in the lab, so that the single cell then come will divide into 2 stages, then into 4 to 8 likewise, and once it is ready, it is some of them are being kept in the liquid nitrogen for the future use, some of the embryo, and most probably one is being placed at a time, so that multiphasic pregnancy can be avoided. So that's all for today. Thank you.